Hello, it's Dmitry again. There we go. Here we go. Okay, I should be fine. Hello, it's Dmitry again. Today we'll be covering the Microbiology chapter for the first aid for the USMLE Step 1 2025 edition. We'll be covering the second half of the chapter today. In the previous live stream or video, we were covering sections 1 through 3. And let me just make sure that I can specify what the sections were. They were, I believe, basic biology. Oh, let me go back to it. They were basic biology. We were covering, let's see. Yeah, basic bacteriology, clinical bacteriology, and mycology. Today we'll be covering some of the bigger, very important sections, which would be the parasitology, right, parasites, virology, systems, and antimicrobials. So, again, I want to remind you that the book that I have on my screen right now that you can see is the 2017 edition. I'll be reading from the 2025 edition. And so there's going to be some variations between the two, but they overall are very similar in terms of content. So just bear that in mind. So let's begin with the parasitology section. Protozoa, GI infections. So we have the organism, the disease, the transmission, the diagnosis, and the treatment. Giardia lamblia causes giardiasis, bloating, flatulence, foul-smelling, non-bloody fatty diarrhea, often seen in campers and hikers. Think fat-rich Ghirardelli chocolates for fatty stools of Giardia. Sure, okay, why not? Transmission. It's spread by cysts in water. Diagnosis would be multinucleated trophozoites. I've heard of trophozoite, I guess. Trophozoite. I always pronounce it trophozoite. Or cysts in stool, antigen detection, PCR. The treatment would be tinidazole, nitazoxanide, or metronidazole. I've always heard it as being nitazoxanide plus metronidazole, but they have a tinidazole here as well. And here's a picture, just a picture you can see A and B for Giardia. Entamoeba histolytica causes amoebiasis, bloody diarrhea, dysentery, liver abscess, anchovy paste exudate, right upper quadrant pain. Histology of colon biopsy shows flask-shaped ulcers. Spread by cysts in the water. Serology, antigen testing, PCR, and/or trophozoites with engulfed RBCs in the cytoplasm, or cysts with up to 4 nuclei in the stool, which you can see, I believe, in picture D for you. Or cysts with up to 4 nuclei in the stool. Entamoeba eats erythrocytes. Okay. Metronidazole is how you treat it. You treat it with metronidazole and paramycin for asymptomatic cysts and cyst passers. What about Cryptosporidium? Cryptosporidium causes severe diarrhea in AIDS patients. You have mild disease, watery diarrhea, in immunocompetent hosts, though. So there's a little bit of difference between how it presents for people who are immunocompromised versus people who are immunocompetent. It's transmitted by ooze, cyst, and water. The ooze cyst on acid-fast stain, antigen detection, PCR. That's how you diagnose. You treat it with prevention, i.e. filtering. Nitazoxanide is how you can treat it as well. Severe disease and or immunocompromised. So again, it might not be necessarily too crazy when you have a person who is immunocompetent, but you would need something more, a little bit something with more, strength to it. Essentially, something like nitazoxanide for somebody who is immunocompromised. What about protozoa, CNS infections? So this is central nervous system infections. What do we think? Toxoplasma gondii. Immunocompetent, so for diseases for Toxoplasma gondii, we have for the immunocompetent people, mononucleosis-like symptoms, negative heterophile antibody tests. And that's important to say, for example, because if you have Epstein-Barr virus, you will have mononucleosis, but you'll be heterophile positive. And so toxo is different, you'll be heterophile negative, but have similar symptoms. Reactivation in AIDS leading to brain abscesses, usually seen in multiple ring-enhancing lesions on MRI. And you can see that in picture A. Congenital toxoplasmosis, classic triad of chorioretinitis, hydrocephalus, and intracranial calcifications. It's transmitted via cysts in meat, most common, ooze cysts in cat feces. It crosses placenta, and pregnant patients should avoid cats. Again, that would make it a torch infection, by the way, because it can be transmitted transplacentally. Diagnosis for Toxoplasma gondii would be serology, biopsy, tachyzoid, and that's going to be a picture in B. PCR of amniotic fluid for possible intrauterine disease. How do you treat it? Sulfadiazine, pyrimethamine, prophylaxis of Bactrim, which is trimethoprim sulfamethoxazole, when CD4 count is less than 100. Now, I want to say something along with that. So, sulfadiazine and pyrimethamine are in fact different drugs than Bactrim. However, trimethoprim has the same mechanism of action as pyrimethamine, and sulfamethoxazole has the same mechanism of action as sulfadiazine. And so basically the combination between the two, sulfadiazine and pyrimethamine, is going to have the same kind of mechanism of actions, the two same mechanisms of action as the Bactrim will have, the trimethoprim sulfamethoxazole. So Toxo is known for using this alternative drug combo, but they do have a very similar set of mechanisms of action. What about Naegleria fowleri? For Naegleria, it causes rapidly fatal meningoencephalitis. Very nasty stuff. It can kill you pretty quickly. It's transmitted by swimming in warm fresh water and enters the CNS through olfactory nerve via the cribriform plate. Diagnosed via amoebas in CSF. Treatment would be amphotericin B and has been effective for a few survivors. Trypanosoma brucei. It causes African sleeping sickness, enlarged lymph nodes, recurring fever due to the antigenic variation, somnolence, and coma. It's spread via tsetse flies. I've heard tsetse flies. It's TSC, tsetse flies, CC flies. A painful bite from one of those flies. Diagnosis would be trypanosomal in the blood smear, as you can see in picture D. And how do you treat it? Suramin for blood-borne disease or melarsoprol for the CNS penetration. I am sure mellow when I'm sleeping. The way I remember it is that I forgot how Suramin's mechanism of action works. I don't know if you need to know that for the exam or not. Maybe you do, maybe you don't. But the way I remember it is called African sleeping sickness. And African sleeping sickness is primarily treated through Suramin. Of course, melarsoprol is very important to do for the CNS stuff, which is relevant. But I think about an African in Suriname. There's an African, if you've been to Suriname, there's people who are, I think in Suriname is next to Guyana. I think technically Suriname is the Dutch colony, so they're going to have people there who are basically black Africans in Suriname. And so I think African sleeping sickness, I think there's an African in, or like I guess it'd be Afro-Surinamese or whatever, Afro-Surinamese. I don't even know what you would call that. In Suriname. So Suramin you treat for African sleeping sickness, i.e. Trypanosoma brucei. Brucei, we'll get on to brucei later on, but brucei. So African sleeping sickness, treat with Suramin, also melarsoprol if there's CNS penetration. What about protozoa for hematologic infections? This is where we're going to get into Plasmodium species. And also Babesia, which actually has some of the similar presentation. So Plasmodium species. My book actually delineates between the different ones, and it's kind of relevant because they do have different cycles and stuff, but it's just a different way of presenting the information. So we're talking the same thing again: organism, disease, transmission, diagnosis, treatment. Plasmodium. Malaria, cyclic fevers, headaches, anemia, splenomegaly, hypoglycemia, and severe disease. Transmission via Anopheles mosquitoes. Diagnosis would be peripheral blood smear. Also allows for identification of the species. The treatment, if sensitive, chloroquine; if resistant, mefloquine, doxycycline, or atovaquone progranil. If it's life-threatening, use intravenous quinine or artesunate. Test for G6PD deficiency, though. Now, what about P. malaria? So Plasmodium malaria specifically, that particular species of Plasmodium. Malariae has a 72-hour fever cycle. It's a Cortan. Diagnosis, trovozoite ring within the RBC. What about P. vivax and P. ovale? Vivax and ovale. They're actually technically a little bit different from each other, but they just kind of consider them the same thing in this book, and I've heard them being conflated before as well. So what about Vivax and ovale? It's a 48-hour fever cycle, tertian, dormant form hypnozoite in the liver. So there's a hypnozoite type that just hangs out in the liver, sort of like in the stasis period. Diagnosis for Vivax and ovale: trophozoites and Schuffner stippling, small red granules within the RBC cytoplasm. How do you treat it? Add primaquine to the target hypnozoites. So primaquine will help knock out those hypnozoites in the liver if you have ovale or Vivax. What about falciparum? Plasmodium falciparum. Disease would be severe, irregular fever pattern. Parasitized RBCs may include capillaries in the brain, cerebral malaria, kidneys, and lungs. Pretty bad stuff. How do you diagnose? Trophozoite ring, headphone shaped within the RBC. And you can see that in the picture, I think. Within the RBC, crescent-shaped gametocytes. Not fun. So just be aware of that, by the way. That when you have P. malaria, 72 hours. When you have P. Vivax or ovale, 48 hours. And when you have falciparum, that's not only is it going to have an irregular fever, it's going to have an irregular fever pattern, basically. So just be aware of that. What about babesia? Babesia is its own little thing. But babesia, fever and hemolytic anemia, predominantly in northeastern and north-central United States, asplenia with increased risk of severe disease due to the inability to clear infected RBCs. So keep that in mind. It has some of the same presentation you might see as you would in malaria, but this is an important distinction. It's not malaria, and it doesn't exist in the same place that malaria would typically find. When you go to, for example, Central Africa, West Africa, whatever, and you have this sort of anemia, splenomegaly, fever, headache, all this kind of stuff, it's not babesia. And if you go to Ohio or something, I'm in East Pennsylvania, and I have I have a fever, I have a hemolytic anemia. Let me give you a hint. It's not malaria. So just keep that in mind. Think about the context of where the patient is getting this if they give you a vignette and ask you what it is. For babesia, for transmission is spread by Ixodes ticks, which is also, by the way, the vector for Borrelia burgdorferi, which causes Lyme disease, and Anaplasma species. So this is another thing, too. When you're reading a question, for example, and they're describing babesia, basically, and you're saying, well, it's asking, what spreads this species of this disease, they're going to have Ixodes ticks on that list of things. And you have to know that it's the same thing as Lyme disease, because you don't want to, psych yourself out and say, I know that can't be the thing because Lyme disease is caused by Ixodes ticks. No. Ixodes does both. Ixodes does Borrelia burgdorferi. Ixodes ticks causes babesia. And apparently it's also related to the Anaplasma species. So just keep that in mind. How is it diagnosed? Ring form, which you can see, I think, in the picture, in a Maltese cross with PCR. So Maltese cross, PCR, and then the ring form. So one of those three things. Treatment, how do you treat babesia? Atovaquone and azithromycin. Protozoa. Others. Okay. So these are going to, again, organism, disease, etc. So visceral infections. Trypanosoma cruzi. This is the other trypanosoma species. We were talking before about Trypanosoma brucei, which we have two different types. I think you have Rhodiens and Gabiens. Those are different places in Africa, Gambia and Rhodesia. Formerly Rhodesia, I guess. In a southern part of Africa. And Gambia is in West Africa. So Trypanosoma cruzi, Chagas disease, dilated cardiomyopathy with apical atrophy, megacolon, megaesophagus. Trypanosoma cruzi causes big problems, predominantly in South America. Unilaterally periorbital swelling, Romagna sign, characteristic of acute stage. This is another thing you can think of just like logically. I'll get into it a bit. I'll just wrap this and I'll just explain to you. Transmission of how do you get Chagas disease? Triatamine insect kissing bug bites and defecates around the mouth or eyes, leading to fecal transmission into the bite site of mucosa. Yeah. So it's not the bite itself, it's the fact that it puts feces in the wound, basically. Diagnosis, trypanomastogote in the blood smear. And you can see that in the picture A. And how do you treat it? Benzonidazole or nifurtimox? So, your thing has a little thing that says benzonidazole, nifurtimox, cruising my bands with a fur coat on. Okay, whatever. Here's the thing with this. It's called Chagas disease. The name is called Trypanosoma cruzi. Cruz is a Spanish word for, I believe, cross, if I'm not mistaken. So it is a Spanish word. If you know, yeah, cross. Okay, I'm not a Spanish expert, but I'm familiar with that. So cruz, like Veracruz, like I guess Ted Cruz, I suppose. Whatever, Santa Cruz. If there's going to be the word cruise, so cruzy, think about it's a Spanish word. So where's it going to be? It's going to be in Latin America. So they say South America. And it also has something called the Romagna sign. It has a little N with a little squiggle on top of it. It's an N, called Romagna sign. So what does that tell you? It's also Spanish. So all those little Spanish things to it, cruzy, Romagna, that kind of thing, all that's connected to it. And that should give you a hint that it's South American. This is in contrast to Trypanosoma brucei, which is called African sleeping sickness, which has names like Gambiennes and Rhodesiennes, which are, I guess those are subspecies of it. And those are going to be in Africa because obviously Gambia is in Africa, for example, and Rhodesia was in Africa. And it's called African sleeping sickness. So that should help you kind of distinguish the two. So just think about the context clues and how these things are connected. It says here about transmission, by the way, my thing says triatamine insect kissing bug bites. Your book says reduvid bugs. I've heard it was reduvid bugs in the past. I never knew they were called triatamine insect. Regardless, though, they are kissing bugs, and you should probably know both names. I would just say, no, it's called a reduvid bug, and also know that it might be called a triatamine bug. The name changes are crazy because it's like, okay, whatever, it's the same thing, but constantly changing everything's name, you just got to know all the names at the same time for the same thing. Leishmania species, visceral leishmaniasis, Kala-Azar, spiking fevers, hepatosplenomegaly, pancytopenia, cutaneous leishmaniasis, skin ulcers, and you can see that they don't show that picture there. Okay. Transmission, sandfly, spread by sandflies. Diagnosis for leishmania, macrophages containing amastigotes. How do you treat it? Amphotericin B, sodium sibogluconate. Okay. What about this is the one STI parasite or protozoa that you should be aware of, which is Trichomonas vaginalis. It causes vaginitis, which is a foul-smelling greenish discharge, itching and burning. Do not confuse with Gardnerella vaginalis. Is a gram-variable bacterium associated with bacterial vaginosis. It's a little bit different from that. It is a transmission and sexual. It cannot exist outside the human because it cannot form cysts. Oh, that's interesting. Its diagnosis through trophozoites, which are motile on wet mount, punctate cervical hemorrhages, strawberry cervix. Kind of gross. Treatment would be metronidazole for patient and partners, prophylaxis, check for STI. And also just keep that in mind that for men, it's not as noticeable necessarily. So you want to make sure that if you're in a situation where this person, presumably a woman, has these symptoms, that any individual that you might be having relations with, if you understand what I'm saying, should also probably get treatment for it because it will likewise probably be present even if they don't notice it. Okay. Where were we? Okay. Nematode routes of infection. Nematode routes of infection, ingested Enterobius oscaris, Toxocara, Trichinella trichuris, Cutaneous strongyloides, Ankylostoma necator. Okay, sorry, so ingested types, so nematode routes of infection. This is nematode routes of infection. The ingested, Enterobius oscaris, Toxocara, Trichinella trichuris. And here's a learning device. You'll get sick if you eat these. Okay. Cutaneous types, strongyloides, Ankylostoma, necator. These will get into your feet from the sand. Okay. Just bear in mind, by the way, adena, which is what I would typically call it. I call that Ankylostoma duodenale and Necator americanus, those are spread through the feet. I don't necessarily know if I guess they say strongyloides is included as well though. So just I guess you can keep that in mind. Bites would be Loa Loa, Onchocerca volvulus, Vaucheria bancrofti. Lay low to avoid getting bitten. Okay, so Loa Loa has its own species of fly that you have to think about. Onchocerca volvulus has its own. Just be aware of, that these are things that can be bit by some sort of insect form, and sometimes it's kind of a specific species. Sometimes, so just look into that if you feel like you need to. Let's talk about nematodes now. Nematodes. I guess we're skipping the immune response to helminths. You can read that if you want to. A lot of eosinophilia, just for reference. Nematodes, roundworms. So fun. Nematodes. Organism, disease, transmission, treatment. So intestinal, nematodes. Enterobius vermicularis, or pinworms. Causes anal pruritus, worse at night. The eggs, which you can see in the picture, I think for A. I'll go down to that. The eggs, visualized via tape test, most common in children 5 to 10 years of age. Transmission is fecal-oral. Its treatment would be bendazoles, like albendazole and mebendazole. Also, you can treat something with parental pemawate as well. What about Ascaris lumbricoides, which is a giant roundworm? May cause obstruction at the ileocecal valve, biliary obstruction, intestinal perforation, migrates from the nose/mouth, migration of the larvae to the alveoli, leading to Loeffler syndrome, pulmonary eosinophilia. Transmission would be fecal-oral, knobby-coated oval eggs seen in feces under microscope. He treated with bendazoles again, that'd be like albendazole or mebendazole. Strongyloides stercoralis, threadworm. Its disease would be GI, like duodenitis, pulmonary, like dry cough, hemoptysis, and cutaneous, like pruritus. So a lot of different things it can affect. Hyperinfection syndrome may be caused by accelerated autoinfection in the immunocompromised. Transmission would be via larvae in soil penetrate the skin. Rabdidiform larvae seen in feces under microscope. Ivermectin or bendazoles. What about Ankylostoma species Necator americanus, hookworms? Cause microcytic anemia by sucking blood from the intestinal wall. The cutaneous larva migrans, which is something you can get from it. Pruritic serpiginous rash. And that's going to be shown in the picture in C. Or I don't think you can see it in your thing, but I have a picture for it, so well. Anyway. We're a serpiginous rash. So how do you get the Ankylostoma species? Larvae penetrate skin from walking barefoot on contaminated beach or /soil. You treat with bendazoles or parental pemawate. Trichinella spiralis. Larvae enter the bloodstream, insist in the striated muscle, leading to myositis, trichinosis, leading to fever, vomiting, nausea, periorbital edema, and myalgia. It's transmitted via undercooked meat, especially pork, or fecal-oral, which is less likely. You treat it with bendazoles. What about Trichuria trichuria, which is whipworm? Often asymptomatic, loose stools, anemia, rectal prolapse in children. It's spread fecal-orally, and you treat it with bendazoles. Just bear in mind, bendazoles are a very common go-to for some of these diseases. For this category of disease, nematodes that are intestinal. What about some of this stuff right here? Toxicara arcanus. Well, it causes visceral larva migrans, migration into the blood leading to inflammation of the liver, eyes, visual impairment, CNS like seizures and coma, and heart, myocarditis. Patients are often asymptomatic though. It's spread fecal-orally, and you treat it with bendazoles. What about Onchocerca volvulus? Causes black skin nodules, river blindness, black sight. Now, Onchocerca volvulus is spread by the female black fly, and it is going to be treated with ivermectin for river blindness. So ivermectin, river blindness. Loa Loa causes swelling in the skin, worm and conjunctiva, nasty stuff. It's spread by deer fly, horse fly, and mango fly, for reference. But it could be from the other two as well. It's treated with diethylcarbamazine. Carbamazine, yeah. What about Vaucheria bancrofti? My thing says here Vaucheria, or maybe say Vaucheria bancrofti, Brugia malayi, as well for some reason. The same thing. Lymphatic filariasis is in the same little camp of things. Lymphatic filariasis, elephantiasis. Look at picture C right there for elephantiasis. Worms invade the lymph nodes, leading to inflammation, lymphedema. Symptoms onset after 9 months to 1 year. It's spread via female mosquito, and it's treated via diethylcarbamazine. Yeah, it looks pretty nasty, doesn't it? Anyway, cestodes, tapeworms. We have Tinea cilium, which has 2 different diseases for that. Let's talk about Tinea cilium that causes intestinal tapeworm. Transmission was ingestion of larvae encysted in undercooked pork, and you treat it with praziquantel. We also have this other disease caused by Tinea cilium called cysticercosis, and also neurocysticercosis, cystic CNS lesion seizures, caused by ingestion of eggs in the food contaminated with human feces. How do you treat it with praziquantel, albendazole for neurocysticercosis? It's really nasty looking. If you've ever seen it, it looks like they have a picture of it in B in your book. My book looks a little bit different. It looks even worse in my book. But look it up right now. Look up neurocysticercosis. Really, really weird. It looks like Swiss cheese kind of brain, which you do not want to have, I would assume. Diphyllobothrium latum. Also, by the way, picture A you have right there for the Tinea cilium. There's 2 different species. There's Tinea saginata, which comes from beef, and Tinea cilium, which comes from pork. They do look a little bit different. The cilium species has little hook teeth, and saginata does not. So if you see a picture of it on the test, just be aware that the cilium species has hook teeth. What about diphyllobothrium? You can really clearly see the teeth on the mouth, you know. What about Diphyllobothrium latum? Causes vitamin B12 deficiency. Diphylloboth12 latum. Okay. Tapeworm competes for B12 in the intestine, leading to megaloblastic anemia. I'd imagine it would also cause some issues with the CNS as well, but that's neither here nor there. Transmission, ingestion of larvae in raw freshwater fish. Treatment would be praziquantel and niclosamide. What about Echinococcus granulosus? It causes a hydatid cyst, eggshell calcification, most commonly in liver and lungs. Cyst rupture can cause anaphylaxis. It's transmitted by ingestion of eggs and food contaminated with dog feces. Sheep are the intermediate host. So yeah, you will find them in sheep, but the real desired host for this species is going to be canines. How do you treat it? Albendazole, surgery for complicated cysts. So there's all the little pictures for you. Okay. We're going to go to trematodes, but I'm going to take a brief break. Okay. Trematodes, flukes. Schistosoma. So schistosoma, we have different species of schistosoma. We have they mention it right down here, so I'll talk about it. So the disease, transmission, and treatment. Disease: liver and spleen enlargement. Shows the picture that you see there. Well, your book is different. So liver and spleen enlargement, fibrosis, inflammation, portal hypertension. The schistosoma mansoni and schistosoma japonicum can both also cause intestinal schistosomiasis, presenting with diarrhea, abdominal pain, iron deficiency, anemia. Okay, chronic infection with schistosoma hematobium, egg with the terminal spine, which is shown in picture B for you, can lead to squamous cell carcinoma of the bladder, painless hematuria, and pulmonary hypertension. So that's the difference between the two. Basically, the other 2 species, basically hematobium is the one that messes with causes hematuria, bladder problems, that kind of thing, bladder cancer. But the other 2 species, mansoni and japonicum, do not do that. They have their own thing going on with the liver. They say here portal hypertension, fibrosis, that kind of thing. Now, there's also a difference between Mansoni and Japonicum. Japonicum has, I think, none or the spine is either small or absent for Japonicum. I think it might just be absent. And in the case of the Mansoni, it's a lateral spine, so it's like a kind of a sticking on the side. For Hematobium, there's a long terminal spine. So let's say you can see the picture right there. The spine is present in picture B, but it's terminal. It's at the end point. In the case of Schistosoma Mansoni, the spine is lateral, so just bear that in mind. How are they transmitted? Snails are intermediate hosts. They cercariae penetrate skin of the humans in contact with the contaminated fresh water, example swimming or bathing. How do you treat them? Praziquantel. What about Clonorchis sinensis? Biliary tract inflammation leading to pigmented gallstones associated with cholangiocarcinoma. You get it transmitted from undercooked fish. You treat it with praziquantel again. Now we're talking about ectoparasites, so I never really went too much into this stuff, I don't think. When I look at sketchy videos, when I have done preparation for this kind of stuff on parasites, they like to focus on all the intestinal stuff, the cestodes, the nematodes, the flukes, plasmodium, so forth. So this is interesting here. Ectoparasites. Cercoptis scabiei, I imagine that'd be scabies. Mites burrow into the stratum corneum and cause scabies. Pruritus, worse at night, and serpiginous burrows, lines, often between fingers and toes. Doesn't sound great. Common in children, crowded populations, jails, nursing homes, transmission through skin-to-skin contact most common, or via fomites. How do you treat it? Permethrin cream, oral ivermectin, washing/drying all clothes/bedding, treat close contacts. What about Pediculus humanus and Phtherus pubis? Blood-sucking lice that cause intense pruritus with associated excoriations, commonly on scalp and neck, head lice, waistband and axilla, body lice, or pubic and perianal regions, pubic lice. Body lice can transmit in Rickettsia prowazeki, epidemic typhus, Borrelia recurrentis, relapsing fever, Bartonella quintana, trench fever. How do you treat it? Pyrethroids, malathion, or ivermectin lotion, and knit combing. Children with head lice can be treated at home without interrupting school attendance. Okay. So basically scabies and lice, not fun. Parasite hints. Well, I have another parasite I want to go over before we go into that. Cimex lectularis, Cimex, C-I-M-E-X, Cimex lectularius, and Cimex hemipterus. Bed bugs. How could we forget, Bed bugs, blood-feeding insects that infest dwellings. Painless bites result in a range of skin reactions, typically pruritic, erythematous papules with central hemorrhagic punctum. A clustered or linear patterns of bites seen upon awakening is suggestive. Diagnosis is confirmed by direct identification of bed bugs in patient's dwelling. Bed bugs can spread among rooms. Cohabitants may exhibit similar symptoms. Infestations can also spread via travelers from infected hotels. The use of unwashed used bedding. Treatment, bites self-resolve within 1 week. Eradication of the infestation is critical. Now we can talk about parasite hints. So associations in the organism. So this is a little bit of a guessing game, so maybe you can just think to yourself, what do you think? Just close your eyes. It's like, who's that Pokemon? Okay. Biliary tract disease, cholangiocarcinoma. What should you think? Clonorchis sinensis. Brain cysts and seizures. What should you think? Tinea solium, neurocysticercosis. Okay, what about hematuria, squamous cell bladder cancer? What should you think? Schistosoma hematobium. Okay, important. Liver hydatid cysts, exposure to infected dogs. What should you think? Echinococcus granulosus. Iron deficiency anemia. What parasite should you think about? They say Ankylostoma necator. What about myalgias, periorbital edema? What should you be thinking about? Trichinella spiralis. So that's like trichinosis. Okay. Nocturnal perianal pruritus. What should you be thinking about? Enterobius. Portal hypertension. When they say enterobius, I mean Enterobius vermicularis, by the way. Portal hypertension. What should you be thinking about? Schistosoma mansoni and Schistosoma japonicum. And what about vitamin B12 deficiency? What kind of thing would you be thinking about for parasites? Dithrobothrium latum. There we go. Oh, did you get all that correct? Hopefully you did, because some of them might be on the exam, potentially. Who knows? Microbiology, virology. This is our next section. We are currently on page 159, and we wrap up on page, I think, 199? I think so. Yes. So we're on 159. We'll wrap up on 199. Oh, wow. This is longer than I thought. What? Okay. Sure. Well, I'll see if we can do this all in one day. If we can, I'll wrap up in sometime in between. I'll try to avoid going longer than 3 hours today, if I can. Viral structure, general features. So we have different kinds of structures. We have naked virus of icosahedral capsid. It's non-enveloped. See the capsid, see the nucleic acid. We have an enveloped virus of icosahedral capsid. The nucleic acid, capsid, lipid bilayer, surface protein. I have this other thing called an enveloped helical capsid. It's a little bit different. It's going to have a helical capsid with viral RNA and a surface protein and some lipid bilayer. And then this is the one that you have in your... Well, you actually don't have this other one. You don't have a bacteriophage. Bacteriophages look different, very unique. You can look it up online. Base plate, spikes, core, helical sheath, collar, nucleic acid capsid. Let's talk about viral genetics. Viral genetics, recombination. Exchange of genes between two chromosomes by crossing over within regions of significant base sequence homology. Reassortment. When viruses with segmented genomes, for example influenza virus, exchange genetic material, for example the 2009 novel H1N1 influenza, a pandemic emerged via complex... For example, the 2009 novel H1N1 influenza, a pandemic emerged via complex viral reassortment of genes from human, swine, and avian viruses, has the potential to cause antigenic shift, reassortment of genome segments. Complementation. When one of two viruses that infect the cell has a mutation that results in a non-functional protein, the non-mutated virus complements the mutated one by making a functional protein that serves both viruses. For example, hepatitis D virus requires the presence of a replicating hepatitis B virus to supply the HBsAg. So that would be like the surface antigen, I believe. The envelope protein for HDV. So basically, it needs complementation for it to actually cause an infection. Phenotypic mixing. Occurs with simultaneous infection of a cell with two viruses. For progeny 1, genome of virus A can be partially or completely coated, forming a pseudovirion with the surface proteins of virus B. Type B protein coat determines the tropism infectivity of the hybrid virus. Progeny from the subsequent infection of a cell by progeny 1 will have a type A coat that is encoded by its type A genetic material. So, virus A and virus B, they create this sort of progeny thing where it has qualities from both A and B, but it has like almost like it has an A phenotype, for example, or has a B phenotype, but then the actual progeny from that will be it has like a B genotype and an A phenotype or something like that. You know, the phenotype is one, but the genotype is the other. And so any sort of subsequent products will all have the other phenotype because it has that genotype. But the early form of it, the first progeny, will have the opposite phenotype, which is interesting. Viral genomes. Naked nucleic acids of most double-stranded DNA viruses, except pox virus and HBV, and positive-strand single-stranded RNA viruses are infectious. Naked nucleic acids of negative-strand single-stranded RNA and double-stranded RNA viruses are not infectious because they lack the required polymerases to replicate. Virions of the negative-strand single-stranded RNA viruses carry RNA-dependent RNA polymerases to transcribe the negative strand to positive. Okay. Where are we here? DNA viruses. all have double-stranded, so this is characteristics in the mnemonic. Okay, so DNA viruses all have double-stranded DNA genomes like our cells, except Parvoviridae, which has a single-stranded DNA. All are linear except papilloma, polyoma, and hepatinoviruses, which are circular. The mnemonic would be part of a virus. Okay. RNA virus. All have single-stranded RNA genomes except Reoviridae, which is double-stranded RNA. So, but again, normally RNA viruses are single-stranded. Normally DNA viruses double-stranded. There's a couple exceptions here and there. DNA viruses, parvovirus is single-stranded DNA, and for RNA, reoviruses are double-stranded RNA. But anyway, back to that. RNA viruses. Positive sense stranded would be, it says here, mRNA, retro, toga, flava, corona, hepa, calissa, picorna viruses. Negative sense stranded would be arena, bunya, paramyxo, orthomyxo, philo, rhabdo. Segmented types would be bunya, orthomyxo, arena, and reo. So let's talk about it. They say repeatovirus for a reovirus. They say, well, at a retro toga party, I drank flavored Corona and ate hippie California pickles. Always bring polymerase or fail replication. So the negative, and then the other one says for segment is a BOR, so BOAR for the segmented types. Always bring polymerase or fail replication for the negative sense stranded types. That's not a bad one, not too terrible. And for the positive sense, they say, while at a retro toga party, I drank flavored Corona and ate hippie California pickles. Okay. Whatever, if that's if that's how you remember, that's how you remember, I guess. Whatever. Viral envelopes. Viral envelopes. Do we have that on here? Viral envelopes. Generally, envelope viruses acquire their envelopes from plasma membrane when they exit from the cell. Exceptions include herpes viruses, which acquire envelopes from the nuclear membrane. Enveloped DNA viruses like herpes virus, hepatinovirus, pox virus have helpful protection. What about DNA viruses? DNA viruses all are icosahedral and replicate in the nucleus except pox virus. Pox is out of the box nucleus. Viral family, envelope, DNA structure, medical importance. So herpes virus, it is enveloped. It is a double-stranded and linear DNA structure, double-stranded linear. It is, its medical importance is you should see herpes virus entry. Okay, fair enough. Pox virus, it is enveloped, double-stranded and linear. It's the largest DNA virus. Smallpox eradicated worldwide by the use of live attenuated vaccine. Makes more sense. Live attenuated vaccine. Cowpox, milkmaid blisters, molluscum contagiosum, which is another kind of pox, flesh-colored papule with central umbilication. Keratinocytes contain molluscum bodies, which you can see in picture. I don't have a picture here, but you get the point. Hepatinovirus. Hepatinovirus envelope would be it's enveloped. It's partially double-stranded and circular. You can get HBV from it. Acute or chronic hepatitis, not a retrovirus, but has reverse transcriptase. It is the only hepatitis virus that is a DNA virus, by the way. What about adenovirus? It is not enveloped. It is double-stranded and linear. It is it causes febrile pharyngitis, which you can see, I think, in the picture there for you. It causes sore throat, acute hemorrhagic cystitis, pneumonia, conjunctivitis, pink eye, gastroenteritis, and myocarditis. Papillomavirus, it is not enveloped, is double-stranded and circular, causes HPV, warts, cancer, cervical, anal, penile, oropharyngeal, serotypes 1, 2, 6, 11 associated with warts, serotypes 16 and 18 associated with cancer, and also 31 and 33 can also cause cancer. Polyomavirus is not enveloped. It is double-stranded and circular. When you think about polyomavirus, there's BK virus and JC virus. JC virus, progressive multifocal leukoencephalopathy, PML, in immunocompromised patients, like in HIV. For BK virus, transplant patients commonly targets the kidneys. JC, junkie cerebrum, BK, bad kidney. And I think for BK you can get hematuria, so just bear that in mind as well. For parvovirus is not enveloped. It is a single-stranded and linear type, smallest DNA virus, parvus meaning small. B19, parvovirus B19 virus, aplastic crises in sickle cell disease, slap cheek rash in children, erythema infectosum, or fifth disease, infects their RBC precursors and endothelial cells, leading to RBC destruction, hydrops fatalis, and death in fetus, pure RBC aplasia, and rheumatoid arthritis-like symptoms in adults. Okay, now we're going to go into herpes virus. I'm going to take a brief... Wait, where are we? Herpes virus, envelopes, where are we? I'm going to take a brief break. Okay, herpes viruses. Herpes viruses. Enveloped, double-stranded, and linear viruses. Recent data suggests both HSV1 and HSV2 can affect both genital and extragenital areas. So here's the thing. I want to go into that a little bit for you right now. This might be a crash out, this might be a tirade, I'm not quite sure. So HSV1 classically is the oral herpes. HSV2 is classically the genital herpes. Here's the thing. Increasingly over time, you're seeing that that's not the case. Because yes, HSV1 has probably been adapted to living in the sort of oral transmission, that kind of thing. And vice versa for HSV2 for anal genital area. Let's increasingly be seeing opposite situations where people have HSV1 and it's, in the genital area and vice versa. The HSV2 in the oral area is less common, but does happen. And there are more and more cases of genital herpes that are caused by HSV1. The reason is because people are putting their mouths around their groin, or usually other person's groin. As a consequence, they're giving people oral herpes without even necessarily having... They're giving people basically herpes through sexual contact without ever actually having genital herpes, because they're giving them oral herpes genitally via, you understand what I'm talking about, okay? No need to be crude, but that's becoming increasingly common when you look at new cases of genital herpes. Increasingly, it's HSV1. Now, it's still largely HSV2, but that you're increasingly seeing that, and because most people have HSV1, most people have oral herpes. It definitely presents a kind of problem if you engage in that kind of behavior. So just bear that in mind. HSV1 is not nearly as nasty as HSV2, but still, you don't really want to have that. So anyway, so herpes simplex virus 1. Respiratory secretions, saliva. Clinical significance. Gingivostomatitis, keratoconjunctivitis, herpes labialis, cold sores, herpetic whitlow on finger, temporal lobe encephalitis, esophagitis, erythema multiforme, responsible for a growing percentage of herpes genitalis. As I said. Notes. Most commonly latent in trigeminal ganglia, most common cause of sporadic encephalitis, can present as an altered mental status, seizures, and or aphasia. Yeah, it's pretty crazy that this very common condition people have, a lot of people have HSV1, can lead to some really, really, really serious problem, which is sporadic encephalitis, which you do not want to have. So HSV2, it's spread via sexual contact, perinatal. Yeah, so it's like, I'm not sure if you could say that torch. It says perinatal. So when you're giving during birth, right? It causes herpes genitalis and neonatal herpes. Most commonly latent in sacral ganglia, Viral meningitis is more common with HSV2 than HSV1. What about varicella zoster virus, which is HSV3? Respiratory is spread. Its route of transmission would be respiratory secretions, contact with fluid from vesicles. It causes varicella zoster, which is chickenpox and shingles, encephalitis, pneumonia, and encephalitis and pneumonia. Most common complication of shingles is post-herpetic neuralgia. Additional notes: latent and dorsal root of trigeminal ganglia, cranial nerve V1 branch involvement can cause herpes zoster ophthalmicus. Okay. What about Epstein-Barr virus, which is HHV4? This is spread via respiratory secretions, saliva. It's also called kissing disease, common in teens and young adults. It causes mononucleosis, which is a fever, hepatosplenomegaly, pharyngitis, and lymphadenopathy, especially posterior cervical nodes. Avoid contact sports until resolution due to risk of splenic rupture. Pretty serious stuff, by the way. Associated with lymphomas, endemic Burkitt lymphoma, nasopharyngeal carcinoma, especially Asian adults, lymphoproliferative disease in transplant patients. It infects B cells through the CD21, must be 21 to drink beer in a bar. You can also remember, this is probably a little bit crude, you can also remember that Epstein's victims were probably under 21, and it could not be at a bar. Okay. Atypical lymphocytes on peripheral blood smear, non-infected B cells, but reactive cytotoxic T cells, positive monospot test, heterophile antibodies detected by agglutination of sheep and or horse RBCs, use of amoxicillin for presumed strep pharyngitis can cause maculopapular rash. So remember, it's heterophile positive, it's monospot positive. We were talking before about something that was not monospot positive, but it had some of the similar symptoms. It was heterophile negative. But remember, Epstein-Barr virus causes mononucleosis, and it is definitely heterophile positive. And that's another way to remember that Epstein probably heterosexual. Now, he was into some other weird stuff, but and he was also a file of something. I guess he'd be an ephebophile. So he was a hetero-ephebophile. So remember, it's heterophile positive. So anyway, cytomegalovirus. He was also, he was a lot of things. Let's figure like that. Cytomegalovirus. So let's move on. Cytomegalovirus, HSV5. Congenital transfusion, sexual contact, saliva, urine, transplant. It can cause mononucleosis, but it's negative monospot test in this case. In immunocompetent patients, infection in immunocompromised, especially in pneumonia in transplant patients, esophagitis, colitis, AIDS retinitis, cytomegalovirus. You get it? I just remember the CMV retinitis is a thing. Hemorrhage, cotton wool exudates, vision loss, congenital CMV. Infected cells have characteristic owl-eye intranuclear inclusions, latent and mononuclear cells. What about human herpes virus 6 and 7? Saliva, roseola infantum, exanthum subitum, high fevers for several days that can cause seizures, followed by diffuse macular rash, starts on trunk, then spreads to the extremities, and usually seen in children less than 2 years of age. Roseola, fever first, rosy rash later, self-limited illness, HSV7 most common cause of roseola. How long is the number of days? They say for several days. Let's just say several days. I think it's like 4 days. I would like to say 4 days. I'm not quite sure. But for several days. You'll have a fever, and then later on you'll have a rash over the body. So human herpes virus 8, HHV8, spread by sexual contact, causes Kaposi sarcoma, neoplasms of endothelial cells seen in HIV/AIDS in transplant patients, dark violaceous plaques or nodules representing the vascular proliferations. It also kind of supposedly resembles a Bartonella henselae infection as well. Can also affect GI tract and lungs. So these are the pictures you can see. So if you want to go compare the pictures, you can look back up to which one was A, B, C, D. A is the keratoconjunctivitis related to HSV1. The cold sores, I think, are B is a cold sore from, again, HSV1. C, for your book, I don't know. Your book has different pictures in some cases, so I can't tell you for sure. I got to go look back at it if you want to. E looks like you have reactivation of herpes zoster. So, okay. H is going to be the owl eye inclusions you would see in CMV. See the owl eyes? That's kind of interesting. What about HSV identification? HSV identification, PCR skin lesions is a test of choice. CSF PCR for herpes encephalitis. Zinc smear, or zinc test, outdated, is a smear of an open skin vesicle to detect multinucleated giant cells. Commonly seen in HSV1 and HSV2 and VZV infection, or at least used to be seen. Intranuclear is eosinophilic Cadre A inclusions also seen in HSV1, HSV2, and VZV. It's kind of interesting. They say in your older book from 2017, they say zinc smear outdated. My book says zinc smear outdated, but your book doesn't say that. It doesn't say it's outdated at all, as far as I can tell. Yeah, it doesn't seem like it. So I wonder when they can start considering that to be an outdated test. Receptors used by viruses. Yeah, the virus and the receptors. So let's talk about it. CMV goes for what? The integrins, heparin sulfate. What about EBV? It goes for CD21. What about HIV? CD4, CXCR4, and then CCR5. And bear in mind, by the way, CD4 I believe is like an early thing it goes for, like CD4 and I think maybe CCR5, not quite sure. But CXCR4, that's like a longer term, like it's like a later stage basically. CXCR4 I believe is later stage stuff. Let me just confirm. What stage of disease does it start attaching It says here, early infection for HIV. The virus predominantly uses CCR5 co-receptor during initial phase. And then it says here, later disease progression. During this phase, often as CD4 count levels decline, the virus mutates to attach to the CXCR4 co-receptor. Okay, so basically CXCR4 is like a later stage thing you would see, while CCR5 is like an earlier thing you might see. What about parvovirus B19? P antigen on RBCs. Rabies attaches to the nicotinic acetylcholine receptor. Rhinovirus is attached to the ICAM1. I came to see the rhino. Okay. That's sure. You could say I used a camera to take a picture of a rhino. Whatever floats your boat. ICAM1. And SARS-CoV-2 attaches to ACE2. Oh, that's another little interesting thing for you. They don't have SARS-CoV-2 on here in your book. Probably because it wasn't a thing back then. I don't think SARS-CoV-2 because that just means COVID-19, right? SARS-CoV-2. SARS-CoV-2. Yeah, I think so. SARS-CoV-2 is the thing that causes COVID-19. Okay. So yeah, they don't have that in your book because it was 2017 when this was made. So let's go back to the next one. Go to the next page to RNA viruses. RNA viruses. It says here, RNA viruses all replicate within the cytoplasm except retrovirus and influenza virus. Retroflu is at a site. Okay. You get it site as in cytoplasm? Anyway, viral family, envelope, RNA structure, capsid symmetry, and a medical importance. So reoviruses. They are not enveloped. They have a double-stranded linear multi-segmented presentation. They cause rotavirus, important cause of diarrhea in young children, may be fatal. What about coronaviruses? They are not enveloped, they are naked. I'm going to say they're naked, okay? Because that's what I've heard it as. Single-stranded positive sense linear viruses. You have poliovirus, which causes you have a polio salk/subin vaccine, IPV, OPV. Echovirus, which causes aseptic meningitis. You have rhinovirus, which causes the common cold. You have Coxsackie virus, which causes aseptic meningitis, herpangina, mouth blisters and fever, hand, foot, and mouth disease, myocarditis, and pericarditis, depending on which type you have. Coxsackie virus A can cause herpangina and hand, foot, mouth disease, but Coxsackie virus B causes the myocarditis. It causes pleurodenia as well. What about HAV, acute viral hepatitis? So remember perch as the different kinds of coronaviruses. Hepavirus is not enveloped, it's naked. Single-stranded linear positive sense virus. It causes HEV. What about caliciviruses, which also include norovirus and norovirus/Norwalk virus? Not enveloped, it's naked. Single-stranded linear positive sense. Causes norovirus, which causes viral gastroenteritis. What about flaviviruses? Flaviviruses are enveloped. They are single-stranded linear positive sense viruses. They cause HCV, yellow fever, dengue fever, West Nile virus, which causes meningoencephalitis, acute asymmetric flaccid paralysis, and they also cause Zika virus. Nasty stuff, you know. What about togaviruses? Togaviruses are enveloped, single-stranded positive sense linear viruses. They cause the crude conditions, which is chikungunya virus, co-infection with dengue virus can occur. Rubella, formerly atogavirus. Okay. Eastern and Western equine encephalitis as well. So basically, chikungunya, rubella, Eastern and Western equine encephalitis. Okay. What about matonaviruses? Matonaviruses are enveloped, single-stranded positive sense linear, causes rubella. So I guess rubella was originally maybe considered to be in the togavirus camp, and now they've made it a matonavirus, which is something a little bit different. Whatever. They say it's formerly a togavirus. Okay. Does your book even have matonaviruses? I don't think so. So that kind of implies exactly what we just said, which is that it used to be considered it's part of the crew, the toga crew, and then they said, oh no, actually it's its own thing, matonavirus. Okay. What about retroviruses? Retroviruses is enveloped, single-stranded positive sense linear. They have reverse transcriptase, HTLV, T-cell leukemia, HIV, AIDS. Okay. Nasty stuff. You don't want to get AIDS. Coronaviruses. It is enveloped, single-stranded positive sense linear virus, causes the common cold, SARS, COVID-19, and MERS. So coronavirus, you said common cold, SARS, and MERS. So there's a little bit of an addition in recent years to that. So, yeah, COVID-19 is included. Orthomyxovirus. It is enveloped, single-stranded, negative sense, linear, multi-segmented influenza virus. What about paramyxoviruses? They are enveloped, single-stranded, negative sense, linear, paramyxovirus, PRM, parainfluenza, which is croup, RSV, which is bronchiolitis in babies, and measles and mumps. Okay. Rhabdovirus. Or mine has pneumoviruses. Pneumovirus is enveloped, single-stranded, negative sense, linear, causes RSV, bronchiolitis in babies. I'm not sure if there's two. Listen, I don't know if there's two different kinds of RSV. I'm not quite sure how that works, but I just have that there as well. Just be aware of that. Rhabdoviruses, they are enveloped, they are single-stranded linear viruses, and they cause rabies. Rhabdovirus. They also have a bullet-shaped to them as well. A lot of these things are helical. Oh, weird. My book doesn't have the specification. Your thing says, oh, helical, icosahedral. I think you should kind of know that a little bit, to be honest with you. But my book, they took that out. I don't know what happened. But anyway, rabies does not have a helical shape. It has a bullet shape to it. They say that's the capsid symmetry. Rabies virus famously has a bullet-shaped capsid, not helical necessarily, but bullet-shaped. Filovirus. Filovirus is enveloped, single-stranded, negative sense linear, causes Ebola/Marburg virus diseases, often fatal. Arenavirus. Arenavirus is enveloped, single-stranded, positive and negative circular multisegmented. It causes LCMV, lymphocytic choriomeningitis virus, Lassa fever encephalitis spread by rodents. What about bunyavirus? Bunyaviruses are enveloped, single-stranded, negative sense, circular, multisegmented, causes California encephalitis, sandfly/Rift Valley fevers, Crimean Congo hemorrhagic fever, and hantavirus, which is a hemorrhagic fever and pneumonia. Delta virus, which is enveloped, is a single-stranded, negative sense, circular. HDV is defective, requires presence of HBV to replicate. That's not a bad way to remember it, sure. So remember, single-stranded, double-stranded, double-stranded. Okay, SSS, yeah, okay. Positive sense, negative sense, arbovirus, arthropod, born, mosquitoes, ticks. Okay. I'm going to go into picornaviruses now. Picornaviruses include poliovirus, echovirus, rhinovirus, Coxsackie-virus, HAV. RNA is translated into one large polypeptide that is cleaved by virus-encoded proteases into functional viral proteins. Poliovirus, echovirus, and Coxsackie-virus are enteroviruses and can cause aseptic viral meningitis. Picornavirus is RNA, picornavirus, small RNA virus, perch on a peak, pico. Okay, sure. Rhinovirus, which is a picornavirus, non-enveloped RNA virus, causes a common cold, greater than 100 serologic types, acid labile destroyed by stomach acid, therefore it does not infect the GI tract, unlike the other picornaviruses which do. Rhino has a runny nose. Remember rhino means nose, like a rhinoplasty? So just bear that in mind as well. Rotavirus, segmented double-stranded RNA virus, a reovirus, most important global cause of infantile gastroenteritis, major cause of acute diarrhea in the United States during winter, especially in daycare centers, kindergartens. Villus destruction with atrophy leads to a decrease in absorption of sodium and loss of potassium. Rotavirus equals right out of the anus. Okay. Sure. If that's how you remember it, I don't know what okay. Rotavirus. CDC recommends routine vaccination of all infants except those of history. It's not, it's just kind of, it's just a little gross. CDC recommends routine vaccination of all infants except those with a history of intussusception, rare adverse effect of rotavirus vaccination, or SCID. I'm taking a brief break. Anyway, let's go back to it. Influenza viruses. Orthomix of viruses, enveloped negative sense single-stranded RNA viruses with segmented genome, contain hemagglutinin, binds to the sialic acid and promotes viral entry, and neuraminidase, promotes progeny virion release. Antigens. Patient at risk for fatal bacterial superinfection, most commonly Staph aureus, strep pneumonia, and H. influenza. Treatment would be supportive plus or minus neuraminidase inhibitors like oseltamivir and zanamivir. Hemagglutinin let the virus in. Neuraminidase sends the virus away. Reformulated vaccine, the flu shot, contains viral strains most likely to appear during the flu season due to the virus's rapid genetic change. There's always issues with vaccination in this context because they just so rapidly evolve. So there is always it's not perfect. It's not perfect. It's not a perfect thing just because we know of how quickly these things evolve over time. Kill viral vaccine is most frequently used. Live attenuated vaccine contains temperature-sensitive mutant that replicates in the nose but not in the lung. Administered intranasally, sudden shift is more deadly than gradual drift. True. The drift is just like, mutations and stuff like that you would see endogenously in that population, but shift is when you actually take up DNA from like a different, separate population. So like swine flu is an example of that, where it starts picking up DNA from like another virus that's not necessarily part of the human influenza virus population. Genetic shift, antigenic shift. So a genetic/antigenic shift. Infection of one cell by two different segmented viruses, example swine influenza and human influenza viruses. Oh, well, speak of the devil. Leading to RNA segmented reassortment, leading to a dramatically different virus, which causes genetic shift, leading to major global outbreaks or pandemics. And the reason is because your body just was not used to that. Now, your body might be familiar with some of the older viruses if it's like, mutated form of something you saw like last flu season. But if you're talking about something that's so radically different because it's from a different population of influenza viruses, well, then it's going to be a lot more susceptible to that kind of infection, and it's going to be a lot more likely to lead to a pandemic. What about genetic and antigenic drift? Random mutation in hemagglutinin, HA, or neuraminidase, NA, genes leading to minor changes in the HA or NA protein drift occur frequently, local seasonal outbreaks, epidemics. So true. What about rubella virus? Rubella virus is a matonavirus, causes rubella, formerly called German 3-day measles, fever, post-auricular and other lymphadenopathy, arthralgias and fine, maculopapular rash that starts on the face and spreads centrifugally to involve trunk and extremities. Causes mild disease in children but serious congenital disease, a torch infection. Congenital rubella findings include classic triad of sensorineural deafness, cataracts, and patent ductus arteriosus. Blueberry muffin appearance may be seen due to the dermal extramedullary hematopoiesis. Okay. What about paramyxovirus? Paramyxovirus causes disease in children. They include those that cause parainfluenza, croup, mumps, measles, RSV, and human metapneumovirus. All subtypes can cause respiratory tract infections, bronchiolitis, pneumonia in infants. All contain surface F fusion protein, which causes respiratory epithelial cells to fuse and form multinucleated cells. Palivizumab, monoclonal antibody against F protein, prevents pneumonia caused by RSV infection in premature infants. Palivizumab for paramyxovirus, RSV prophylaxis in preemies. What about acute laryngotracheobronchitis, or CRU? So, also called CRU, caused by parainfluenza viruses. Virus membrane contains hemagglutinin, binds the sialic acid and promotes viral entry, and neuraminidase promotes progeny virion release. Antigens results in a seal-like barking cough and an inspiratory stridor. Narrowing of the upper trachea and subglottis leads to a characteristic steeple sign on X-ray, as you can see in the picture. Now, your book also says severe CRU can cause pulsus paradoxus secondary to upper airway obstruction. What about measles, rubeola virus? Usual presentation involves prodromal fever with cough, coryza, and conjunctivitis, and then eventually Koplik spots, bright red spots with blue-white center on buccal mucosa. You need to know it's inside the mouth, by the way. It is in the mouth, not somewhere else. Followed by 1 to 2 days later by a maculopapular rash that starts at the head/neck and spreads downward. Lymphadenitis with Finkel-D, giant cells, fused lymphocytes in a background of pericortical hyperplasia, possible sequelae, subacute sclerosing panencephalitis, SSPE, personality changes, dementia, autonomic dysfunction, death occurs years later. Okay. Encephalitis, 1 in 1,000, symptoms appear within a few days of the rash, and then giant cell pneumonia, rare except in immunosuppressed people. So my thing says, what are the 4 C's of measles? Your thing says 3, but mine says 4. The 4 are cough, coryza, conjunctivitis, and also Koplik spots. Yes, Koplik starts with a K, but they're saying, hey, we're just going to, just kind of worm that way in. Work it in as the fourth C. Vitamin A supplementation can reduce morbidity and mortality from measles, particularly malnourished children. Pneumonia, vitamin A supplementation, by the way, vitamin A. Pneumonia is the most common cause of measles-associated death in children. Okay. So this is in coryza as well. So cough, coryza, conjunctivitis. I just want to make sure coryza is the acute inflammation of the mucous membranes lining the nasal cavity. So it's like, it says synonymous with acute rhinitis. So like nasal mucus, basically. Mumps virus, uncommon due to the effectiveness of the MMR vaccine. Symptoms are parotitis, as you can see in the picture, orchitis, inflammation of the testes, aseptic meningitis, and pancreatitis. Can cause sterility, especially after puberty. I've heard before from people that taught my classes, or taught a class for me, that mumps is unlikely to cause infertility, but it technically can, but it's unlikely. Just be aware that it can technically cause infertility. Even if it's not necessarily super common, it can happen. Mumps makes your parotid glands and testes as big as pom-poms. Okay. And then pom as in like parotitis, orchitis, meningitis, pancreatitis. Now we're going to go into arboviruses. Remember, arboviruses are arthropod-borne. So we're talking about chikungunya and dengue, so I don't know where we have that. Oh boy. We were on a roll for a few minutes there, and now we're not, Ooh, let's see what we can do. You know what? Let's leave it on the slide right here. Let's leave it right here, and then I'll just read off the viruses for you, okay? We have yellow fever on here. Did we have yellow fever on here? We have Ebola. Arbovirus is transmitted by Aedes mosquitoes. So we have chikungunya virus and dengue fever virus. So we have virus type. So let's talk about chikungunya. Chikungunya is an alpha virus / toga virus. It causes high fever, maculopapular rash, headache, lymphadenopathy, and inflammatory polyarthritis. Arthralgias are most commonly reported versus dengue. So arthralgias are more commonly reported for chikungunya. Joint swelling is highly specific for chikungunya. Thrombocytopenia, leukopenia, and hemorrhagic manifestations are less common. You diagnose chikungunya using RT-PCR and serology. Treatment would be supportive steroids or disease-modifying antirheumatic drugs was a DSMARD? Well, Estimard, D-M-A-R-D, yeah, okay. For chronic arthritis, prevention would be minimize mosquito exposure. No vaccine currently available for chikungunya. What about dengue fever? Dengue fever is a flavivirus, causes dengue fever, rash, headache, myalgias, arthralgias, retroorbital pain, neutropenia, dengue hemorrhagic fever, which causes dengue fever plus bleeding and plasma leakage due to the severe thrombocytopenia and RBC perturbations, most common if infected with a different serotype after initial infection due to the antibody-dependent enhancement of the disease. Interesting. May progress to dengue shock syndrome with plasma leakage leading to circulatory collapse, which I would imagine is not good. Diagnosis would be through RT-PCR serology, just like chikungunya. Now, dengue fever treatment would be supportive in intravascular volume repletion or blood transfusion if severe shock is caused. And you prevent dengue fever via live recombinant vaccine, which is available, derived from a yellow fever virus black backbone with insertion of genes for the envelope and pre-membrane proteins of dengue virus. So what about yellow fever? Yellow fever is a flavivirus, also known as an arbovirus, transmitted by the Aedes mosquito bites. Virus has a monkey or human reservoir. Flavi means yellow or jaundice. Symptoms is high fever, black vomitus, jaundice, hemorrhage, backache, may see Councilman bodies, eosinophilic apoptotic globules on liver biopsy. Live attenuated vaccine recommended for travelers to endemic areas. Zika virus, which you have right here, is a flavivirus most commonly transmitted by the Aedes mosquito bites. Causes conjunctivitis, low-grade pyrexia, and itchy rash in 20% of cases. Outbreaks more common in tropical and subtropical climates. May be complicated by Guillain-Barré syndrome. Supportive care, no definitive treatment. Diagnosed with RT-PCR and serology. Sexual and vertical transmission occurs. In pregnancy, can lead to miscarriage or congenital Zika syndrome. Brain imaging shows ventriculomegaly, subcortical calcifications. Clinical features in the affected newborn include microcephaly, ocular anomalies, motor abnormalities, spasticity, and seizures. What about rabies virus? Rabies virus, bullet-shaped virus. Thank you. Now they call it bullet-shaped. They were calling it helical before, but now they say, oh yeah, bullet-shaped. Of course it's bullet-shaped. Bullet-shaped virus, negri body cytoplasmic inclusions, commonly found in Purkinje cells of cerebellum and hippocampal neurons. Rabies has a long incubation period, weeks to months, before symptom onset. Post-exposure prophylaxis is wound cleaning plus immunization with killed vaccine and rabies immunoglobulin. Example of passive active immunity. Travels to the CNS by migrating in a retrograde fashion via dynein motors up nerve axons after binding to acetylcholine receptors. Progression of disease includes fever, malaise, leading to agitation, photophobia, and hydrophobia, and hypersalivation, which leads to paralysis, coma, and death. So, once you start seeing things like photophobia and stuff like that, you're basically going to die. Like, you're done. Sorry, you're cooked. It's over. You're chopped. Too bad. So you really want to get it treated basically before you even start seeing symptoms. If you start seeing really serious stuff, like you start seeing agitation, photophobia, hydrophobia, yeah, you're just going to die. There's not really anything you can do at that point. Maybe they could try to treat you, but you're just going to likely die. So. Get treated once you get bit by something like a bat or a dog. You have suspicion you get bit by a bat. Get treated before you start seeing symptoms, if you want to get better and don't want to die. Infection more commonly from bats, raccoons, and skunk bites than from dog bites in the United States. Although bear in mind when we say in the United States, that's a very important caveat because in a lot of other countries, dogs are a huge perpetrator because they simply don't have them vaccinated for it. The United States has rabies vaccines for dogs, and a good reason for why that should be the case is because otherwise they would be giving people rabies all the time. So, in a developing country, you could say, where they do not give the dogs rabies vaccines, you're going to see possibly them being a real meaningful vector for rabies, which is not fun. Anyway, aerosol transmission for bat K is also possible, but again, usually it's from bites. Aerosol transmission sounds horrifying, though. What about Ebola virus? Ebola virus is a filovirus following an incubation period of up to 21 days, presents with abrupt onset of flu-like symptoms, diarrhea/vomiting, high fever, myalgia, can progress to DIC, diffuse hemorrhage, and shock. Diagnosed with RT-PCR within 48 hours of the symptom onset, high mortality rate. Transmission requires direct contact with bodily fluids, fomites, including dead bodies, infected bats or primates like apes and monkeys, primates, parentheses, apes/monkeys. Like, I didn't know what primates we were talking about. The ones, I was thinking about like the primates that are not apes and monkeys, I mean, there is a technically different term. Primate is a thing that can be used referring to something else. It refers to like a particular, I think, like ecclesiastical office, like a primate is like a certain kind of patriarch in the Catholic Church or something like that. But we all know you're talking about monkeys, okay? Let's be honest, okay? It could be found in primates, like monkeys. Oh, I didn't know. Okay, I was thinking of I was thinking of the other primate, right? Whatever, man. High incidence. It's like saying felines, parentheses, cats. Oh, I wasn't sure. High incidence of healthcare associated infection. Supportive care, no definitive treatment. Vaccination of contacts, strict isolation of infected individuals, and barrier practices for healthcare workers are key to preventing transmission. What about severe acute respiratory syndrome, coronavirus 2? You don't have it in your book, but I have it in mine because there was no such thing as a COVID-19 apparently back in 2017. I don't know what that's about. Trying to drop in the ball in 2017. What's going on? It would have been nice to know about this beforehand, right? Anyway, SARS-CoV-2 is a positive sense single-stranded RNA virus that causes caused the COVID-19 pandemic. Spread is via the respiratory droplets, aerosols, and fomites. Varying symptoms include fever, myalgia, headache, nasal congestion, sneezing, cough, sore throat, nausea, diarrhea, anosmia, and dysgeusia. It's dysgeusia. Severe cases may lead to complications like pneumonia, hypercoagulability, DVT, PE, stroke, myocardial injury, neurologic sequelae, shock, organ failure, and death. Diagnosed by NAAT, RT-PCR, viral antigen test, RAT, rapid, accessible, less sensitive than NAAT. Host cell entry occurs by attachment of the viral spike protein to ACE2 receptor on the cell membranes, highly expressed in the lungs, intestines, heart, and kidneys. Vaccines, mRNA, and viral vector vaccines induce immunity by presenting the spike protein to the immune system, leading to the anti-spike protein antibodies. Spike S protein shown and this is a little figure here. Spike S protein, there's a lipid bilayer, there's a membrane protein M, envelope protein E, and then the helical capsid with the viral RNA. Remember, he has a helical capsid to it. Okay, we're going to go to hepatitis viruses. I'm going to take a brief break. Anyway, hepatitis viruses. We're back. Okay, hepatitis viruses. Signs and symptoms of all hepatitis viruses are episodes of fever, jaundice, elevated ALT and AST. Here's a learning device. Naked viruses, HAV and HEV. Lack an envelope and are not destroyed by the gut. The valves hit your bowels. Okay, and then that'd be BCD. BCD hits your bowels, and they're the valve. Well, sorry, A, and E. Those are the vowels. My mistake. The valves hit your bowels. They are not destroyed by the gut. They lack an envelope. HBV DNA polymerase has a DNA and RNA dependent activities. Upon entry into nucleus, the polymerase completes the partial double-stranded DNA. Host RNA polymerase transcribes mRNA from the viral DNA to make the viral proteins. The DNA polymerase then reverse transcribes the viral RNA into DNA, which is the genome of the progeny virus. HCV lacks 3' to 5' exonuclease activity, leading to no proofreading ability, leading to antigenic variation of the HCV envelope and proteins. HCV envelope proteins. The host antibody production lags behind production of new mutant strains of HCV. And so here's a little chart for you. You can go and take a snippet of that for your notes if you want to. So these are the different viruses, A, B, C, D, and E. So HAV, family would be the RNA picornaviruses, spread fecal orally, shellfish, traveler's daycare. Incubation is short, weeks. Clinical course is acute and self-limiting for adults and asymptomatic in children. The prognosis is good. The hepatocellular carcinoma risk is none. They say no. Liver biopsy, because it's acute, it's short. liver biopsy, hepatocyte swelling, monocyte infiltration, Councilman bodies. And the notes for HAV, absent, no carrier state. What about HBV? B as in boy. It's in the DNA hepatinovirus. It's a DNA hepatinovirus. I also want to say that hepatinovirus, yes, correct. It's transmitted parenterally through blood, sexual, bedroom, or perinatal, birthing. Incubation would be long, months. The clinical course is initially like serum sickness, fever, arthralgias, rash, may progress to carcinoma. Prognosis, adults mostly full resolution. For neonates, worse prognosis. There is a risk for hepatocellular carcinoma. For liver biopsy, granular eosinophilic ground glass appearance due to the accumulation of surface antigen within infected hepatocytes. Cytotoxic T cell mediates damage. Notes for HBV, carrier state is common. What about HCV? HCV is an RNA flavivirus, primarily blood injection drug use post-transfusion. Incubation is long, may progress to cirrhosis or carcinoma. The majority develop stable chronic hepatitis C. HCC risk, hepatocellular carcinoma risk is present, yes. For biopsy, lymphoid aggregates with focal areas of macrovesicular steatosis. And for notes, carrier state very common. What about HDV? RNA delta virus, also part of the viroid family. But delta virus, parenteral, spread by parenterally, sexually, perinatally. Is incubation period superinfection, HDV after HBV short, co-infection, HDV with HBV long. Similar to HBV, prognosis, superinfection is a worse prognosis than the co-infection. Co-infection is not as bad, superinfection is worse. Remember, the superinfection is when you have HDV after HBV, while a co-infection is when you have it at the same time. So when it's happening at the same time, it's co-infection not as bad. If it's happening after you already had HBV, which is a superinfection, that's pretty bad. Prognosis, superinfection, worse prognosis. Is there a hepatocellular carcinoma risk? Yes. The biopsy would be similar to HBV. And for notes for HDV, defective virus depends on HBV surface antigen, HBsAg, code for entry into the hepatocytes. The HEV, RNA hepavirus, fecal oral, especially waterborne, is a short incubation period, fulminant hepatitis in expectant pregnant patients, high mortality in pregnant patients. There's no hepatocellular carcinoma risk, patchy necrosis. Notes would be enteric epidemic in parts of Asia and Africa, the Middle East, no carrier state. And a way I remember it would be like this. A&E, as I said before, lacks an envelope, and they are not destroyed in the gut. They are typically acute infections that do not typically have a chronic state, and they are spread fecal orally. That's A&E. Fecal oral, non-enveloped, can survive the stomach, typically acute, not usually a chronic state, not necessarily a particularly high risk for carcinoma. B, C, and D are not the same. B, C, and D are different than A and E. B, C, and D are sexually transmitted. They are chronic typically, and they can cause carcinoma. They can cause hepatocellular carcinoma. So A and E are similar to each other. Non-enveloped, in the stomach, they can survive the intestines. They're acute. They are not chronic. B, C, D, chronic, sexually transmitted, and they are going to be at risk for hepatocellular carcinoma. What about extrahepatic manifestations of hepatitis B and C? Let's talk about B, hepatitis B. It can cause aplastic anemia, and for the renal, it can cause membranous glomerulonephritis, more so than membranoproliferative glomerulonephritis, but it can cause those. For vascular, it can cause polyarteritis nodosa, so just be aware of that. What about hep C, hepatitis C? It can essential mixed cytocryoglobulinemia, elevated risk B cell NHL. Was that NHL? What is that? I want to say leukemia. NHL. Hep C. It says here non-Hodgkin lymphoma. How can I be so blind to that? NHL, non-Hodgkin lymphoma. So back hepatitis C, essential mixed cryoglobulinemia, elevated risk for B-cell non-Hodgkin lymphoma, ITP, which is to make sure I'm not saying this incorrectly. ITP. Is that immune thrombocytic purpura? It is immune thrombocytic purpura. Thrombocytopenic purpura. Okay. And autoimmune hemolytic anemia. So Hep C can cause all those issues. ITP, NHL, hemolytic anemia, cryoglobulinemia. Now, for the renal, Hep C can cause membranoproliferative glomerulonephritis more so than membranous glomerulonephritis. Vascular, it can cause leukocytoclastic vasculitis. Dermatologic, Hep C can cause sporadic porphyria, cutaneous tarda, and lichen planus. And the endocrine, Hep C can cause elevated risk for diabetes mellitus and autoimmune hypothyroidism. Okay. What about more? This is more and more hepatitis stuff. Hepatitis serologic markers. So, anti-HAV IgM. IgM antibody to HAV, hepatitis A, best test to detect acute hepatitis A. Anti-HAV IgG. IgG antibody indicates prior HAV infection and or prior vaccination, protects against infection. HBsAg, which is going to be the surface antigen. Antigen found on the surface of HBV indicates hepatitis B infection. Anti-HBs, antibody to HBs antigen, indicates immunity to hepatitis B due to vaccination or recovery from the infection. HBcAg, which is the surface antigen for hepatitis C. HBc antigen. Antigen associated with the core of HBV. So core, sorry, I'm wrong. HBcAg would be core antigen for HBV. Anti-HBc. Antibody to the HBc antigen. IgM equals acute recent infection. IgG equals prior exposure to or chronic infection. IgM anti-HBc may be the sole positive marker of infection during a window period. HBeAg, HBe antigen, secreted by the infected hepatocyte into circulation, not part of the immature HBV virion, indicates an active viral replication and therefore a high transmissibility and poorer prognosis. So that's important to note that the HBe antigen, the E, is referring to a not part of the mature HBV virion, which means that it's probably actively going on, because if it wouldn't have these sort of immature antigens hanging around if it wasn't actively going through production, basically. Anti-HBe, antibody to HBe antigen indicates low transmissibility. There's a little chart right there for you to see how these things line up. So you can see if you have XYZ thing, you can tell if it's incubation period, acute infection, window period, recovery period, chronic infection. Chronic infection, you have the surface antigen is present, the core antigen is present, the core antibody is present, I should say, and the E antigen is present, and that tells you there's a chronic infection where there's a high infectivity. Only the acute infection and the chronic infection have HBe antigen present. Everything else does not have that. Chronic infection, low infectivity as well, and then immunized state. Immunized state, you only have anti-HBs present. You only have the surface antibody present. Everything else is absent. All the other antibodies are not present, and none of the antigens are present when you have an immunization. Because if you actually had a recovery, you would have all the antibodies should be present for that. For a recovery stage, you should have all the antibodies, but only the surface antibody will be present if you're immunized, essentially. I'm going to take a brief break. Okay. We are on HIV. So we can look at that if you want to. I think they will probably have a question potentially on hepatitis, so definitely know your stuff on that stuff. Don't go too crazy with it, but you should know it. Remember for these board exams, bugs and drugs, know your microbiology and know your pharmacology. Very important stuff. Because even if you know your pathology, oh, I know about this disease, I know about that disease, et cetera, et cetera, et cetera. If you don't know the drugs for how to treat it and how they work and all that other kind of stuff, all the complications they're in, you're going to run into a lot of problems. And if you don't know the bacteria and the viruses, et cetera, that might cause diseases and how they operate and how they work and how you treat them and so forth, you're going to run into some problems as well. So these are very important topics. HIV. Envelope proteins. So a little picture right here. Let me talk about it right there. Picture. You see the envelope proteins acquired through the budding from the host cell plasma membrane. GP120. That's the docking glycoprotein GP120. The GP41. That's the transmembrane glycoprotein. We also have this thing called the P17 matrix protein. Lipid envelope, P24 capsid protein, the reverse transcriptase. So let's talk about HIV. HIV is a diploid genome, 2 molecules of RNA. The 3 structural genes protein coded for are ENV, ONV, GP120 and GP41 formed from the cleavage of GP160 to form the envelope glycoproteins, GP120 attachment from the host CD4 positive T cell, GP41, 41 fusion and entry, GAG, P24 and P17 capsid and matrix proteins respectively, POL, reverse transcriptase, integrase, protease, RIP, POL, POL. Whatever floats your boat. Reverse transcriptase synthesizes double-stranded DNA from the genomic RNA. Double-stranded DNA integrates into the host genome. Virus binds to the CD4 as well as the co-receptor, either CCR5 on macrophages, early infection, or the CXCR4 on T cells in late infection. Remember, macrophages early on at CCR5, and then late-stage CXCR4 on T cells. Homozygous CCR5 mutation leads to immunity. Heterozygous CCR5 mutation leads to slow course. HIV diagnosis. HIV 1/2 antibody antigen immunoassays detect viral P24 antigen capsid protein and IgG and or IgM to HIV 1/2. Use for diagnosis. Very high sensitivity/specificity, but may miss early HIV disease if tested within first 2 weeks of infection. A positive screening test is followed by a confirmatory HIV 1/2 differentiation immunoassay. HIV RNA tests detect elevated HIV RNA and can be qualitative or quantitative. NAAT is qualitative and is a sensitive method to detect HIV viremia in antibody-negative patients. Viral load tests like RT-PCR are quantitative and determine the amount of viral RNA in the plasma. They're used to monitor the response to the treatment and transmissibility. Western blot tests are no longer recommended by the CDC for confirmatory testing. HIV 1/2 antibody antigen testing is not recommended in babies with suspected HIV due to the maternally transferred antibody. Use HIV viral load instead. AIDS diagnosis, less than 200 CD4 count cells. Okay, so the CD4 count is less than 200. Normal would be 500 to 1500 cells per cubic millimeter. Or HIV positive with AIDS-defining condition like pneumocystis pneumonia. There's a little diagram that I have that you do not have. It says here, HSV diagnosis, HIV 1/2 antibodies and P24 antigen combination immunoassay. If it's negative, well then negative for HIV1 and HIV2 antibodies and P24 antigen. If it's positive, then HIV/HIV 1/2, or HSV1, HSV2 antibody differentiation immunoassay. And you can have HIV1 positive or HIV2 negative, HIV1 infection. If it's HIV1 negative but HIV2 positive, then it's an HIV2 infection. If they're both positive, then HIV1 and HIV2 infection. That doesn't sound particularly fun. If they're both negative, then you have to give the person an HIV1 NAT, and that'll help determine whether or not you have an HIV1 NAT positive for an acute HIV1 infection or an HIV1 NAT negative for a negative HIV1 situation. So basically, if you test positive for the original test, and you have an HIV1/HIV2 positive antibody differentiation immunoassay, but you can't figure out whether it's 1 or 2, then you have to do an HIV1 NAT to figure whether or not you have 1 infection or not. Anyway. Here's a little chart for you. Time course of the untreated HIV infection. I do not have that in my book. And so that might mean they don't consider it to be relevant anymore. I don't know. If you want to look at that and figure out what's going on with that, be my guest. Looks cool, doesn't it? Okay. Interesting. Death. You see the little thing at the end there for the HIV RNA? Goes up and up and up. Death. Does not sound good. Anyway. Common diseases of HIV positive adults. Common diseases of HIV positive adults. Decrease in the CD4 positive cell count leading to a reactivation of past infections like TB, HSV, shingles, dissemination of bacterial infections and fungal infections like coccidiomycoses, and non-Hodgkin lymphomas. So we have pathogen presentation and findings. For a CD4 count less than 500, what do we think? Candida albicans, oral thrush, you would find scribable white plaque, pseudohyphae on microscopy. For EBV, you would have oral hairy leukoplakia. You would have unscribable white plaque on lateral tongue. So if it's unscribable, it's EBV. If it's scribable, then it's Candida. HHV8, Kaposi sarcoma, localized cutaneous disease, perivascular spindle cells invading and forming the vascular tumors on cystology. HPV, squamous cell carcinoma at sites of sexual contact, most commonly anus, cervix, and oropharynx. Mycobacterium tuberculosis, increased risk of reactivation of latent TB infection. What about CD4 counts that are less than 200? So now we're really actually in HIV territory now. Like we're actually in AIDS territory, I should say. They have HIV regardless in this case, but what if we're in actual AIDS territory? Less than 200. Histoplasma capsulatum. Fever, weight loss, fatigue, cough, dyspnea, nausea, vomiting, diarrhea. You would have oval yeast cells within macrophages. What about HIV? Dementia, HIV-associated nephropathy, cerebral atrophy on neuroimaging. JC virus reactivation, progressive multifocal leukoencephalopathy. That would be PML, by the way. Is a non-enhancing areas of demyelination on MRI. What about HHV8? Kaposi sarcoma, disseminated disease, pulmonary GI, lymphatic. What about PJP, pneumocystis jirovecii? It causes pneumocystis pneumonia. That's a classic disease you would have if you actually have AIDS. You have ground glass opacities on chest imaging. Bear in mind there's other things that could cause ground glass appearance other than PJP, but that's one of the things that can cause it. What if we're talking about CD4 count that's less than 100? Doesn't sound fun. Bartonella species, bacillary angiomatosis, multiple red to purple papules or nodules, biopsy with neutrophilic inflammation. Candida albicans will have esophagitis, with white plaques on endoscopy, yeast and pseudohyphae on biopsy. Cytomegalovirus CMV causes colitis, retinitis, esophagitis, encephalitis, and pneumonitis, creep. Linear ulcers on endoscopy, conmole spots on endoscopy. Biopsy reveals cells with intranuclear owl-eye inclusion bodies. What about Cryptococcus neoformans? It causes meningitis. You would have encapsulated yeast on India ink stain on the capsular antigen, which is positive. So Cryptococcus neoformans would be encapsulated yeast on India ink stain or capsular antigen positive. What about Cryptosporidium species? You have chronic watery diarrhea with acid-fast oocysts in stool. Epstein-Barr virus, B-cell lymphoma with non-Hodgkin lymphoma, CNS lymphoma. CNS lymphoma leading to ring-enhancing lesions, I would imagine, maybe solitary like versus toxoplasma, which would be multiple. So toxoplasma would have multiple ring-enhancing lesions. For EBV, you would only have like a singular ring-enhancing lesion. What about Mycobacterium avium complex, or Mycobacterium intracellulare? Those would be non-specific systemic symptoms: fever, night sweats, weight loss, diarrhea, or superficial lymphadenitis. Most common in CD4 counts that are less than 50. Exactly. When you have it, when it's less than 50, it's probably MAC. If it's more than 50, it's probably, I think, TB. Because it sounds similar to each other. MAC and TB sound similar to each other in terms of how they present themselves. Non-specific systemic symptoms, fever, night sweats, weight loss, right? You think, oh, maybe that's TB. Well, if it's less than 50, it's probably MAC, but if it's more than 50, it's probably TB. Toxoplasma gondii. I don't even think they mentioned TB in here. TB reactivation can happen, I think, with HIV. Anyway, Toxoplasma gondii causes brain abscesses, multiple ring-enhancing lesions on MRI. Keep in mind that if it was only a single one, it might be more likely to be EBV. But if it's multiple, it's probably toxo. Now let's talk about prions. Now we're on page 174. I made a decision that I will not complete the chapter today. I will complete the chapter in the next live stream. We will be wrapping up on this part on, what section are we on, technically speaking? Systems? We're in virology right now. We're in virology. We'll cover the systems section in the next video. Because I know that if I wrap up now, I will finish at around 2 hours-ish, that kind of thing. If I were to do the other portion of this chapter, I'll probably wrap up in another 2 hours or so, and I don't exactly want to do that per se. So I'll just wrap up now, and I'll go into it the next time. So prions. Prions, diseases are caused by conversion of a normal, predominantly alpha helical protein termed prion protein, PRPC, to a beta-pleated form, PRPSC, which is transmissible via the CNS-related tissue, iatrogenic CJD. Or food contaminated by BSE-infected animal products, variant CJD. PRPSC resists protease degradation and facilitates the conversion of still more PRPC to PRPSC, resistant to the standard sterilizing procedures including standard autoclaving. Accumulation of PRPSC results in spongiform encephalopathy and dementia, ataxia, sterile myoclonus, and death. It is not fun. Prion degeneration is very nasty. And it's super freaky too, because it's not actually a living. It's not like this is in the microbiology chapter, sure, but it's not actually like an organism. It's not a virus, and it's not a microbe in the way that we think of a bacteria or a parasite. It is a protein that's misfolded, and it creates this whole chain reaction that will kill you eventually. Anyway, so we different kinds: Creutzfeldt-Jakob disease, CJD, rapidly progressive dementia, typically sporadic, some familial forms. You have bovine spongiform encephalopathy, which is also called mad cow disease, which was in the UK, the United Kingdom at one point, and Kuru, acquired prion disease noted in tribal populations practicing human cannibalism. They don't mention where that is. It's in Papua New Guinea. Tribal populations that practice human cannibalism. First off, versus what, the non-human cannibalism? What else are you cannibalizing exactly if you're a person? Whatever. But, just bear that in mind. Kuru, BSE, mad cow disease, right? And then Creutzfeldt-Jakob disease. Anyway, so that concludes that section, the virology section of the chapter today. And we will be wrapping up the last part, I guess it would be part 3, for the microbiology chapter in our next stream. Probably, I guess today is Monday, so probably Tuesday. Again, I didn't necessarily want to have it to be a 3-part live stream for this one chapter. However, it is one of the longer chapters of the series, and I don't exactly want to be here for a 7-hour live stream to have it all in one go. So I will probably get back to you tomorrow. If you watch this video and watched it to the entire way, hopefully it was a good use to you. Otherwise, thank you for watching, and have a great rest of your day. Thank you.